Thank you for visiting my talk on the Science of Consciousness conference online. I would like to talk about a unifying theory of consciousness, which connects the underlying biochemical mechanisms in living systems. Before we get into that, I'd like to introduce myself. My name is Rajnish Khanna. I'm a plant photobiologist. In the past several years, I've been working in the agriculture and food industries. In 2015, I presented this TEDx talk in Livermore, California, on a local food application called Terra Local that we are developing, and we are about to launch it in the next few weeks. I-Cultiver is a company that I started about 4 or 5 years ago. It connects research and analytical needs of the agriculture industry with academia. ClassLight is a software developed by one of my colleagues, Greg Asner, and it uses satellite images to monitor deforestation. So just a little few of the other things that I am involved with, but my interest is also in the hard problem of consciousness. And consciousness is viewed by religion and science, I think, in slightly different ways. But then we find that psychedelics connect the two viewpoints in some way, where science meets religion in accepting in some way that it is giving some sort of a spiritual or some sort of a experience that is also connected with meditation and so forth. And there are many studies with brain scans that are showing images of how the brain is reacting under psychedelic influence. And of course, so all of this is occurring in biology, which is taking place in the physical universe, in space-time, and quantum mechanics is playing a role. So to understand a problem like this, I think we need to be more inclusive of other sciences. The mathematics, physics, and cosmology, which have done wonderful jobs in describing the universe, are more descriptive. I think we need some more experiential sciences, such as philosophy and biology, to participate in understanding how consciousness works. So we will take a step directly into this presentation by looking at light. As a photobiologist, I view light as a source of information. So light is packaged with information. It carries information about direction, duration, intensity, and even color, the quality. And the color, as you can see here, I'm showing the visible light, which is a very small spectrum of the entire radiation that's coming from the sun. But the radiation is packed with the information of frequency and wavelength and energy. So this information is what plants are capable, for example, in interpreting or unpacking. Now, here we are looking at a model organism called Arabidopsis. These are seedlings of this plant Arabidopsis, grown in darkness for 4 days, or in continuous light, red light, or far red light for 4 days. So you can see how differently these seedlings grow in these different environments. In darkness, the seedlings are much longer, taller, and they have a very prominent hook, and the cotyledons have not opened. So we'll be focusing on these two organs, hypocotyls and cotyledons. Hypocotyls are these organs that are elongating the hypocotyl or the seedling, and with a prominent hook at the top. And you can see when these seedlings are grown in light, the hypocotyls are shorter, and the cotyledons are more open and expanded. And this happens also in the far red light. However, in far red light, we can see that there is no greening. So red light, which is needed for photosynthesis, can start the chlorophyll biosynthesis pathway and cause greening, which does not occur in far red light. So we can see clear differences in developmental patterns of these. We can look at a little bit closer at this program of dark growth and light growth. Columbia is the what we call the wild type version of this Arabidopsis, which is the normal response to the light signal, let's say, in this case. And we are looking closely at the cotyledons. So you can see that in darkness, in all cases, these three images that we are showing, we can see there's a hook and the cotyledons have not expanded. When we look at the wild type, in red light, the cotyledons are well expanded, opened. In far red light, they are not as expanded, but they are open. Now, phi A and phi B represent two different photoreceptors. These are photoreceptors which will perceive red or far red light depending on the environment of quality. If there is more red light versus less red light, which can happen under shaded conditions, but we won't get into that. But simply to show that if we mutate one of these photoreceptors, for example, phytochrome A, which is phi A, we can see that in red light, you get expanded cotyledons, which means that the plant is able to sense red light, even in the absence of phi A. However, it cannot sense far red light because under far red light, it grows as if it was in darkness. So clearly then phytochrome A is involved in sensing far red light. Similarly, phytochrome B, which is involved amongst other photoreceptors in sensing red light. So when phytochrome B is missing, the activities of phytochrome C, D, and E, which are the other phytochromes, is ongoing. So phytochrome B mutants, which lack the function of phytochrome B, will not expand their cotyledons as much as the wild type will in red light. Whereas in far red light, phytochrome B mutants are able to perform as equally as well as the wild type Columbia seedlings. So in other words, by mutating one gene, we can alter the developmental program. So now this connects us with an external signal, such as light, and its influence on the organisms and the genetic makeup of the organism that plays an important role. So these images are from one of my publications in 2003, and I was involved with this another publication in 2004. So we can see how sensitive plants and other living organisms are to an external signal. So let's take a little bit closer look so that we can understand the underlying biochemical mechanisms that are involved in sensing signals that are coming to us from outside. So let's look at darkness, and you won't have to remember the names of these proteins or genes. There is no test at the end. But we will just use the names so that we can orient ourselves within the signaling pathway. So here in darkness, COP1 SPA1 complex, which is a very important complex, which needs to be active for the dark adaptation, which we call scotomorphogenesis. So the dark elongation of hypocortical, which is known also as etiolation, as we saw up here. This process is called etiolation or scotomorphogenesis. And then in light, we get photomorphogenesis. So now here we're looking at the etiolation or scotomorphogenesis process, where COP1 and SPA1 complex play an important role by suppressing the activity of this protein called Hi5. And another protein, BBX32, also suppresses the activity of Hi5. The genes mentioned or proteins mentioned with the asterisk, Hi5 and PIF5, are the ones that I was involved in identifying several years ago. But this process then keeps the Hi5 program off. So this off switch for this Hi5 program will keep the other system, which is the PIF1, 3, 4, and 5 system, active, which leads to transcription or regulation of downstream genes, leading to scotomorphogenesis, or this growth pattern in darkness. So let's turn the lights on. Now when I turn the lights on, as we discussed before, there are different wavelengths of light, the far red, and then blue and UV, and they're all sensed by different photoreceptors. We discussed phytochrome A and phytochrome B. For blue light, there are cryptochrome, phototropin, Zeitlupe, and for UV, there is UVR8. So these different specific, very sophisticated sensory mechanisms in plants, here we are talking about Arabidopsis, will then activate the photomorphogenesis pathway or development in light pathway. And to do that, they will suppress the COP1 SPA1 complex, activity of that complex, and inhibit the PIF system with all the PIF proteins which promote scotomorphogenesis. So by turning these pathways off, then the Hi5 pathway can be turned on. And Hi5 works together with BBX20, 21, and 22 to promote photomorphogenesis. So this is the development in light, and as you can see, these seedlings, these have been grown in light, and they have shorter hypocotyls, and cotyledons are open. And so this whole system, which has been divided in dark, different mechanism, and in light, different mechanism, shows us that how the developmental program is entirely different in the two light or dark regimes. So what happens if we take a gene out? So what is the consequence of a loss of function? Let's say of this COP1. COP1 is, as I mentioned, an important interface which promotes scotomorphogenesis by suppressing Hi5. So if we take COP1 out, if we mutate it, if we lose the function of COP1, what we expect is that its effect on PIF system will be gone, and also its suppression of the Hi5 will be gone. So which means that we have turned off the off switch. There is no off switch now. What if we now take the light signal away? If there is no light signal and there is no off switch, obviously there is no regulation of the off switch, then one would expect that this system will be active, Hi5 will be active, and it will promote photomorphogenesis, which means even in the absence of light, we will get this phenotype or the morphology of a light-grown seedling. And indeed, that is what we get. So we take COP1, which is known as constitutively photomorphogenic. It was identified in 1992 by one of my colleagues. It was a very big discovery, I would say, in plant biology in 1992. And it showed us how the photomorphogenesis can proceed when both the light signal and its off switch, COP1, are both absent. Right? So to think about it in a way, I would suggest if you have a car which is parked on a downhill slope and your foot is on the brakes. So if you remove the brake, you don't need gas. You are just going to roll down the hill by just removing the brake. So similarly, the photomorphogenesis program is sort of already programmed in the seedling, and it's being turned off by COP1. When you remove COP1, you don't need light, you don't need gas to go down that hill, and you will get photomorphogenesis, which also suggests that the survival of the seedling may depend upon light, because ultimately, like we saw earlier, for chlorophyll biosynthesis, you need photosynthetically active light. So light is needed to survive. So the seedling growing in the dark will grow as long as there is nutrients within the seeds or to support in the environment. But if the nutrients are depleted, then the plant cannot photosynthesize and will not make it. However, we just saw that the growth, a different program of growth can occur without light. So this is an important distinction. Now let's look at just the light pathway itself. So the light pathway has been conserved from ancient times. Even in prokaryotes, we can find photoreceptors, primitive forms of photoreceptors. Photoreceptors are present in animals and humans, and also obviously in plants. And the downstream mechanisms are very similar. They involve molecular, chemical, and genetic regulation, transcription through transcription factors. There is protein stability. Stability of proteins is involved. Phosphorylation and other types of protein modifications are involved, and this is conserved throughout evolution. So this system developed very early on how to perceive the signal which is outside, how it is perceived and transmitted to cause changes. So now let's try a thought experiment. In this thought experiment, we'll think about this development occurring in Minkowski spacetime. Minkowski spacetime obviously takes into account not just the X, Y, and Z dimensions, but also the fourth dimension of time. So, and that's really where development is occurring. So at a very localized micro space, which has these coordinates of X, Y, and Z, the development is occurring over moments of time. So we take my example for here. I'm sitting here There is this, if I walk back that way, that will be, let's say, y-axis. And if I'm over here, this is x-axis. But if I climb up one of these trees, that's z-axis. But all of these things will be occurring in moments of time. As I'm talking, time is passing. So there is another movement of space through this time. And so the thought experiment involves how does development occur through time, and does that have anything to do with evolution or with the fate of the biological system? So let's look at this. We know there are several factors involved in driving or sustaining life on Earth. Light, of course, is one. Gravity, which is constant mostly, except for maybe some altitude and latitude differences. Air as well, oxygen and carbon dioxide for plants is important. Temperature, water, and mineral nutrients. And if you look at all of these, light is represented with this symbol lambda. And it is quite variable depending on where you are. Quantity of light, quality of light, direction of light, even duration of light can be quite variable. Gravity can be somewhat variable, air is variable somewhat, and temperature, water, and mineral nutrients can show quite a high degree of variation. So with this variable factors, if we introduce those into this diagram now, we can see that light, gravity, and these factors shown in green are actually specific ones that our planet Earth provides. Air, temperature, water, and nutrients, which can be highly variable. So gravity is also a feature of Earth. So the only one factor that we are talking about is non-earthly or is coming from the sun. All these other factors are provided by the nature of Earth. So light is an external signal, but if light is not there, the etiolation process will move towards the direction of time. And if light is there, then photomorphogenesis. And the variation of light during the day, which is shown over here, with a peak of intensity, let's say in midday. So light varies quite a bit, but then what drives the movement of growth and development through time? So here I'm proposing that there is a need for another signal, and this signal is hidden. We don't have the ability to measure it yet. And this hidden signal I call Aleph, with the symbol Aleph. And now we will try to look at why is there a need for this signal, and I will try to show a plausible link where this signal may be perceived by living organisms. So let's add, just for the sake of this talk, let's add it in our table. We say a proposed signal, which is Aleph, written with this symbol. It is undetected. I would say that it is present constantly. And I wouldn't get into this aspect of why I think it is similar to light, but not detected. But it's out of the scope of this talk. But I think that this will be similar in many qualities, such as with light. So what happens in darkness? That is the question that we are going to address. So in darkness, we have COP1 SPA1 complex, like I mentioned before. And what I'm proposing is that Aleph is an external signal, which is present in the absence of light. It's present all the time, but when light is present, light dominates. So the light signaling pathway inhibits or suppresses the effects of or influence of Aleph. Aleph is present constitutively and constantly. It's a constant. So Aleph is acting in the absence of light, and it's moving development through the moments of time without having any of the other factors. So it's driving development to its fullest potential without the influence of any of other factors' presence. Okay, so growth through time is primed to change its developmental pattern in the presence of favorable conditions, which means that Aleph's role is to prime growth and development so that it's prepared for the other factors which are required for the survival of life. Okay, so let's continue the thought experiment. If this is true, then Aleph maintains growth and developmental programs in space-time to facilitate the organism's survival during adaptive evolution. So it must have played a very fundamental role in what we understand about evolution. And as Herbert Spencer stated, survival of the fittest, this is basically a growth and developmental program that is now adapting to the environment that it is in to maximize or to achieve nutrient inputs. So now if you go back to the example of the downhill car, if you remove your brakes, on a flat surface, your car will not move anywhere. But it's only when your car is on the downhill surface, when you remove your brakes, then it will continue to move downwards. Okay, so this is interesting. I think obviously the more commonly accepted reason for evolution to work is that our genetics is programmed as such that we are genetically programmed to move continuously in developmental programs, and when we run into a challenging time, millions and millions of times one mutation or several mutations occur, and that they allow us to then overcome the challenge, and we have evolved. If we think about the downward hill again, this is equal to your car coming down and finding a gas station. So if there's a gas station, you can put the gas in and you can move on. So what I'm proposing is that gas station itself is pretty complex. So the evolution is co-occurring in many different forms. There are systems that are being developed through evolution, and those systems then are adapting living organisms to deal with their environment or to meet the requirements for the sustainability of life in their environment. So I think this complexity that we find in evolution, which is very hard to explain, and obviously there are people who think pro and con in these terms, but I would say that there is a need for a driver. That hill needs to be there for the developmental program to run its course in a slightly directed way. So this idea is not that far-fetched, I would say, because we already have an example. Light is an external signal, which is clearly influencing growth and development. And 96%, nearly 96% of our universe, matter and energy, we don't know what it is. It is unknown. So it's not such a big leap to think that there is another external signal which is acting like light, and it's influencing growth and development in the absence of light. That is simply what the proposal is. And to draw it a little bit further, I wanted to find, is there a conserved ancient pathway? Just like light signaling is ancient, is there another ancient signaling pathway that we can think of where this signal might be acting? And I found one. I think shikimic acid pathway, or the shikimate pathway, is a very good candidate for this. And the shikimate pathway is active in prokaryotes, fungi, algae, and plants. And actually in plants, it only occurs in plastids. So one can argue that it also has a prokaryotic origin. But the shikimate pathway is involved in biosynthesis of aromatic amino acids like phenylalanine, tyrosine, tryptophan, and all the psychedelic molecules that have psychedelic effects on human brain. They're all derived from one or more of these amino acids, especially tryptophan. So let's come back to that in a second, but let's look at the plant itself first. So in the plant, the shikimate pathway is making tryptophan in the plastid. Then the tryptophan can take multiple paths to make the phytohormone Oxen. Oxen counteractively acts with light to regulate growth and development. And it's primarily involved in cell elongation, so making things longer in opposition to light. And this is very important not only in the scotomorphogenesis or the hypocortical elongation, but also in shade avoidance that I mentioned briefly earlier. So the tryptophan pathway is working to synthesize Oxen in plants. We already know that light acts on this particular mechanism over here, the SUR2 gene, to suppress or reduce the amount of Oxen that is being produced, so that light can slow down cell elongation. So light will enhance the activity away from the biosynthesis of Oxen towards this IGs but we won't get into all of that. And it also has other regulatory mechanisms that reduce the biosynthesis of oxen. So in my proposal, what I suggest is that this is only half of the story. Then the other half is the hidden signal, which I call Aleph, and that it acts through a etoreceptor, which is not known. So the hidden signal, we have not been able to detect it yet. And then it will act through a similar mechanism where there'll be an etoreceptor. Most likely we know what, we have sequenced it and we are aware that this molecule exists, but we don't know its function. So this etoreceptor will exist, and that etoreceptor will regulate an increase in the biosynthesis of oxen. So in the absence of light, there will be an external signal that's driving regular growth and development, priming the organism to achieve maximal inputs that are required for survival. So that is the idea, and that is the reason for Aleph and etoreceptor. So in this thought experiment, we discussed that there exists a hidden signal named Aleph. It drives localized space-time through moments of time, and the name Aleph is being used here just as a placeholder to suggest that there is a possibility of another signal. Now, as I mentioned, 96% of our universe is still unknown. I don't want to yet link this to dark energy or what the signal might be. Simply to say that Aleph is a potential signal and a placeholder for this influencing mechanism on growth and development. And more work is required to link it to some of the other possible mechanisms that we are finding through other approaches in physics, mathematics, and cosmology. And so this signal is perceived by a receptor, which I call etoreceptor, which is named after this etiolation, which is the elongation growth. So a photoreceptor, which is perceiving light. Similarly, there is going to be an etoreceptor, which will be receiving this signal and then transmitting its information to regulate gene expression and other biochemical pathways to guide development towards the other factors that might exist in the given environment. So I also suggested that this pathway or this signal will be acting through tryptophan pathway as at least one of the pathways. There might be additional pathways which also requires more research. But I think that Aleph would act on the tryptophan pathway. So linking this all of this back to consciousness or at least our human brain, we can see that tryptophan pathway is also important in human brains, and it's needed for the synthesis of serotonin, melatonin, and niacin. As you know that the receptors in our brain which are targeted by serotonin are the same receptors that psychedelics act on. So there are similarities, I would say, between all living organisms. And like I mentioned, with the prokaryotic systems, shikimate pathway actually originated in prokaryotes. So the shikimate pathway is active in all living organisms, and there seems to be a missing signal and a missing receptor. With that, I would like to point out that we are also doing a workshop on plant mushrooms, medicine, and consciousness. And this workshop will be open starting September 14th. It'll be online. And we have two talks, one by Dan Horner, one by me. And then we will have a great discussion with Dennis McKenna and Paul Stamets on why do plants synthesize molecules with neuropsychoactive properties. And I highly recommend watching this because both Dennis and Paul brought in some recent data, and it was really a pleasure to hear their thoughts on why plants make these substances and how we can benefit from these plants. So with that, I'd like to end my talk. And if you would like to reach me, here is my email address. You can visit my websites. And thank you very much for listening.